Okay, now here is an overall diagram of cell transport. If you notice, we've got it split up with active transport and, in red, passive transport. Let's take a look first on the right at passive transport. Passive transport, we have the movement of water with osmosis. Here, water can pass through the phospholipid bilayer kind of on its own. It's a very small particle. Then we have the idea of diffusion. Now, you've got simple diffusion, again, which is that movement across the cell membrane on its own, not through a protein. It's simple. So it passes through on its own from an area of higher concentration to an area of lower concentration. Facilitated diffusion is going to involve a protein. It's facilitated. If you facilitate something, you help it. So a protein is going to help it pass through, and we call those proteins ion channels because ions like potassium, sodium, etc. can pass through it. So again, if it's facilitated, it must be helped out. Facilitate is a verb. It's helped out, and it comes through those proteins. The thing is, it's still going down the concentration gradient. Now, if you want to go against the concentration gradient, we've got to move towards the green area of the diagram, which is active transport. In active transport, we need energy. Requires ATP. We're going against the current. We're going against the concentration gradient. So here, if material enters a cell, we have what we call endocytosis. Endo meaning in. If we have material exiting the cell, we have exocytosis. Now, two main ones for endocytosis are phagocytosis and pinocytosis. Now, phagocytosis is kind of like when an amoeba engulfs a paramecium, kind of takes it in. The kind of joking example we could say is if, grandma reaches out and gives you this gigantic bear hug, kind of takes you in, but this time she actually absorbs you and engulfs you. Kind of disturbing. Pinocytosis is kind of if a little pin opening happens, okay, and it's able to take smaller material in. So phagocytosis, it completely engulfs it like an amoeba, and pinocytosis, think pin, think small opening. Okay, it's important to take a look at the tonicity chart here and understand what's going on in this diagram. Basically, you've got three different situations here. You have an isotonic solution that a cell is placed in, hypotonic with a round cell, and hypertonic solution. Iso is a balance of the black salt, which is the solute, and the blue circles, which is the water. It's balanced, therefore, it goes in and out equally. The water will flow in and out equally. Here, the hypotonic solution, you've got more water particles on the outside. It's got low salt. Hypo means low. Low salt in the solution. Remember, you're looking at the solution, which is this outside area. Pretend this is like a beaker. Okay? Pretend this is a beaker. You can just draw that in red right here. And inside the cell is placed. That solution, a lot of water, hypo, very low amounts of salt, those black particles. Tons of salt inside. Therefore, the water is going to flow in. It's going to go from high concentration of water to low concentration of water. It flows towards the higher concentration of salt. The opposite is hypertonic. So in a hypertonic situation, you've got a lot of salt, a low amount of water in the solution. Hypertonic, high solute, again, in the solution. So here's your beaker, the cell is placed in it. You can see there's a ton of salt in the solution. You're always looking at the solution on the outside. the water is in high concentration inside, it always moves from high concentration to low, and it goes in the direction of the solute, so it flows outward. Now, the question you'd ask yourself is, well, what happens to those cells? So, what happens? If you look further down, you can see plasmolysis, or plasmolysized, that's the word, the cell shrivels up. So here, you've got a shriveled up cell at the bottom. Down in here, in a hypotonic, the water rushes in, so it's actually going to possibly swell up or burst. In a plant cell, it's going to go out towards the cell wall. In isotonic, it stays the same. So again, hypertonic, the water rushes out, so it shrivels up like a raisin. In hypotonic, it's going to get really big and possibly burst, almost like a balloon. In isotonic, it's going to stay the same. The cell wall, which is made of cellulose, maintains the plant cell structure, so although the cytoplasm and the cell membrane is going to shrivel, the cell wall will stay structured. Although it expands in hypotonic, it's not going to burst, and here it stays the same. So again, this is an important chart to understand. Okay, here we have your main cell transport diagram. If you notice over here to the left, you've got a very small particle. As you move towards the right, you've got larger red particle. As you move to the right, you've got a larger green particle. Okay, and over here, you can see that we've reversed it with a large dark green square particle. Okay, and what you could see is that in terms of diffusion, simple diffusion, small particles like this on the left can pass through the phospholipid bilayer, just moving down the concentration gradient. From area of higher concentration, you can notice that on the far right over here, they show that with different colored shapes, down the concentration gradient to lower concentration. So here, that small particle passes right through. Now, in a bigger particle, it's still going to go down the concentration gradient, but it needs a channel to go through. So it's channel mediated with a protein. So here we have, in orange, a protein. Then we've got a larger particle here, and in this case, another type of protein needs to help it get through the phospholipid bilayer. All of this is passive. It moves down the concentration gradient from an area of higher concentration to an area of lower concentration. In active transport, if a particle needs to go against the concentration gradient, it needs to move through a protein with energy in the form of ATP. In this case, this gate can open up, close up, and spit the large green square up the other end. So again, here we reiterate the idea that the smaller particles here, here, and here can travel through the phospholipid bilayer, but only one of them can really get through the bilayer on its own through simple diffusion. It simply diffuses through. That'd be like a water molecule. So here, this small one can go right through, pass through that phospholipid bilayer. The larger particles have to go through either a channel, as in this one, or they may need to be mediated by a carrier protein, where the gateway opens up here. Here's the gate right in here. And that gateway opens up, accepts that large green particle, comes in, kind of spits it out the other side. These are all examples of passive transport. Now, if I just erase this, now active transport is we need to go against the concentration gradient. Almost like trying to paddle upstream. In order to paddle upstream, we would really need to use a ton of energy. So that's why you've got in purple here, it's written energy. You go against the concentration gradient, you have to use energy in the form of ATP. Here we're able to use that carrier protein to, in that gateway right there, grab onto the square particle, and with energy it closes and opens up the other side, out in here. Again, that's going from lower concentration to higher concentration, which seems unnatural. It doesn't go from high to low, which is passive. Here we have to use energy in active transport to go against the concentration gradient. Again, the analogy is it's like going and paddling upstream. So don't forget, you have to use energy. Another example of active transport is a sodium-potassium pump. There needs to maintain a balance between sodium and potassium on the outside and the inside of the cell. So in this case, these charged particles, sodium ions or potassium ions, sodium ions are actually pumped out of the cell. And here you can see them with the arrow going outward. Potassium ions, on the other hand, are pumped into the cell. Using ATP, we're able to do this. If you notice the shape of the protein right here in this ion channel, there's actually this nice curved area that we'll just color in green. This nice little curves right in here, a nice little round curve, fits perfectly the sodium. If you also notice that there is a square shape right in here that will fit the square potassiums. So it's kind of like a key where the sodiums go in, they'll be released out. The potassium on the outside will come in, they'll be released inside. So it's that kind of custom fit. And again, it's very mechanical. Anything mechanical like this is probably going to need energy going against the concentration gradient. Again, that's why it's active transport. Another example of active transport is the contractile vacuole. Here you see it in a paramecium diagram. It's that star-shaped structure right here in the middle. It's able to fill with water and then contract using energy to spit the water out. The paramecium, which is an example of a protist, doesn't want too much water coming into it because it'll explode. So it needs to take the water in and then spit it out. Here you can actually see a microscopic image of the paramecium, and that star-shaped structure takes water in, spits it out. Anything that's moving that actively probably is active transport and takes energy. Exocytosis versus endocytosis. Here in this diagram, you've got a situation where you can actually see endocytosis, endo meaning in, where food particles here are kind of taken in. Specifically, this is a form of endocytosis called pinocytosis, which we'll get to later, where like a little pinhole pops in there. Food particles come in, a vesicle is formed around it, but then you see a lysosome digesting it. Well, it's digested, and now we need to get the waste out. So, if we need to get that waste out here, we do something called exocytosis, exo out. So in this case, a vesicle will come towards the surface and it will be moved out of the cell. Okay, so again, endo in, exo out. So here we have some real examples. These are photos taken with transfer electron microscopes. So here we have an amoeba in the upper right in blue, and it's engulfing another organism here. You can see the pseudopods. These are false feet that are reaching around, and eventually it's going to take it all in and kind of engulf it. We can take a look. It's going to kind of bring it all in. The pseudopod's going to come completely around it, and it's actually going to become part of this amoeba, and then digestive enzymes will digest it. Now down here, we've got a white blood cell, which is part of our immune system, very important. Got red blood cells, and then these purple dashed lines in here, little dots. Those are bacteria. You can actually see them getting engulfed right in that space. You can see them right in here, in this area, getting engulfed. This is all examples of phagocytosis. And of course the funny cartoon up there with the poor little bacterium or paramecium that is crying out or thinking to itself, oh no, while the pseudopods absorb it. A little cartoon amoeba. Here we have pinocytosis. So in this case, an organism is not going to reach out with pseudopods, but the particles or the targeted molecules, as you see up in here, are going to come on in, and receptor proteins are going to accept them. A small little cleft is going to form in here in the second stage. And then what's going to happen is it's actually going to bring it in and form a coated vesicle. Later, it can be absorbed for energy. So this is pinocytosis. It's nice to think of it as like a pinhole that forms. The interesting thing is that it actually forms a vesicle to surround it. Down at the bottom is actual images of this happening in nature.